happy to have you here. And yeah, we started recording. So this will all be available on the website as well as slides and tutorials. So make sure to check that. And yeah, today, so excited to introduce Sarah Johnson, who's a second-year computational biology PhD student in the Moorjani lab. She attended the University of Washington for undergrad, where she studied molecular, cellular, and developmental biology. Prior to grad school, she worked at Adaptive Biotechnologies, where she analyzed T and B cell receptor dynamics. And she is broadly interested in human genetics and evolution, and her current research is focused on methods for population genetics of Neanderthal and human DNA from ancient sediment samples. So she's got a lot of cool expertise, and she's going to teach us some ways of using ggplot for biology discovery. So very excited, and please join me in welcoming Sarah. And now.
Okay.
You got it?
Yeah, I think I got it.
Oh, that is
yeah,
I am.
Okay. Okay, great. Thank you for the introduction, and thanks for having me. I'm excited to be here. And today I wanted to talk about R's ggplot function, and specifically how to use that for biological data analysis and visualization. So I'll kind of talk about what this package is, and then I'll have like a presentation kind of on background of it, and then I have an interactive like Google Colab notebook that we can work through. And like Monica said, all of this will be posted on the website. So if you don't have your laptop today, you can always work through that later if you just want to see some examples. And with that, I'll start with a big question, which is, what is ggplot? So ggplot is a package that's a part of the tidyverse, which is like a group of packages within R. So it's a part of the R programming language, and then its own package. And I did say the package name is ggplot2. I'm just going to talk about ggplot, but when you're actually loading it, it'll be ggplot2, is why it says 2. And if you want to know more about the tidyverse, Monica had a great skill seminar presentation on this last year, and that's also online. But you can use this package without knowing the tidyverse. And hopefully everything we're doing today, everyone will be able to follow along, even if you don't have extensive R knowledge. And so ggplot2, if you just Google what is ggplot2 or ggplot, this is the like kind of official line, which is that it's a system for declaratively creating graphics based on this book, The Grammar of Graphics, which I think more clear version of what this is it's just a plotting package that's really flexible and customizable, and it's data frame based. And there is a grammar to it. And today I'll kind of be talking about the grammar and how you understand it and build up to these plots. And learning that can be like a bit of a challenge. It's harder than initially plotting, but once you've learned that grammar, it does become really flexible and easy to customize. And there's a lot of power within it. So that's what it is. And I kind of wanted to just show a few examples of plots that I have made in my time, and all of them have been made in ggplot. Not so you understand all these plots, but just to kind of show you the different ways that you can plot things and visualize things, and the different areas that I've plotted to maybe help you think of ways that this could apply to your own research. So one thing I've done is just look for Neanderthal ancestry within the genome, and you can visualize it across samples, or you can visualize it across the length of the genome. I've conducted simulations, and so then I plotted the results of all the simulations. When I was working at the biotech company, I was looking at immune repertoire. So you could look at the immune repertoire composition across individuals. You can track things over time. So here I was tracking T cells over time. And then finally, like I've done some case control studies. So these are just kind of a random collection of the potential plots you can make. And all of these are made in ggplot. So within this package, there's a lot of flexibility. And the overall goals for today, we're not going to be making a bunch of very beautiful plots, but I really want to introduce kind of the building blocks and the grammar of this, so that hopefully you can leave this knowing how to start making your own plots and understand how they're set up, so that then you can go on and make your own like highly customized plots. We'll also talk about some of the flexibility, and then I have 2 different biological data sets that, depending on how much time we have, we can plot those and look at some real examples. Oh yeah, and again, this is a follow-up to Monica's. So all of this is online. If you have more questions about just the tidyverse in general or R, she has a really good background introduction. So with that, I thought I'd just start by actually talking about the layers of a ggplot. So this is it's a kind of different way of plotting in that everything has layers, and there's different layers. So the first layer that I think is probably the most important are these geomes. So they're called geome, and then they're geometric mapping. And it specifies the shape to be drawn. And I think a lot of this will make more sense when we look at examples. But some examples of geomes are geome point, which is how you would specify a scatter plot, or geome line, which would add a line to your plot. So this is really how you're adding the things that you see on a plot onto your plot. There's lots of examples that we'll talk about later. So you always have to have a geome. And then you need an aesthetic. So AES is their abbreviation. And this is an aesthetic mapping. So it basically will map each point of the geome onto something in your data frame. So this is where you'll specify what your X and Y coordinates are. And you can also adjust the size of each point, the color of each point. So this is ways that you can customize this and then also specify your axes. You can also add on a theme. So this is getting to be a bit more visual. This is where you can change the text size, the font, the color. You can rotate the plot, you can add plot ticks, and you can add lines. So this is where you can really kind of customize how it looks once you have the basics of your plot built with the geomes and the aesthetics. You can additionally add labels, which is always a great thing to do. It will give you default ones, but you can add in your own label. So label your x-axis, label your y-axis, give it a title. You can facet it, which again, I'll show you examples, but this will create panels of plots. So if you don't want to plot everything on one giant plot, if you want to break it into pieces, you can use a facet to do that. So that's all the things we're going to be talking about. What does that actually look like? So I mentioned that this is a data frame based plotting method. And so the input to every plot is going to be a data frame, which is basically a table like you'd see in Excel, but that is just stored in R as a data frame. And you always need an x-axis column, and you almost always need a y-axis column as well. It's like one exception that I'll show you. But in general, you need your axes, and these will be within your data frame. And so how you actually start building your plot is you call the ggplot function. And then the first argument will be the data frame. So just do data equals data frame. And then you'll specify the mapping with the aesthetics. So this is where you'll say the x-axis is going to be this column, which here I'm calling x column. And the y-axis will be the y column. So this is that mapping I said, where you're actually giving it the points that you're using. And if you put it in this order, you don't actually have to say data equals and mapping equals. So in all my examples, the first argument will always be the data frame, and the next argument will always be this aesthetic mapping. Yeah. And so what's an example of this? There's this iris data frame that I'll be looking at within the notebook. But what this has is it'll have multiple flowers, the species, and then multiple measurements from each of those flowers. And so if you wanted to create a plot from this data set, and the data frame was named iris, you would put that in as your data frame, and then you'd specify X is this column species, and then Y, here we're going to plot the sepal length. You just put in that column name. But importantly, this will not yet create a plot because I mentioned that there's geomes, and so you have to add a geome on top. So this is saying, these are the axes, this is the data, doesn't yet know how to actually create a plot. So that's where you have to add a geome. So we'll have our intro setting up the plot, and then you can add a geome point. And so this will add on scatter points. So now onto those mappings that we've created, we'll add on points and we can see the points. This is not necessarily the best way to map this data, but this is the first example that we'll start at. So to make anything show up, you add on the geome. And then from there, oh yeah, so these are all of the different geomes, and I'll show you examples of all of these. We just looked at geome point, which is creating a scatter plot. There's also lines. And once you've looked at this enough, the names actually start being very intuitive. So you want a bar graph, you type bar. You want a box plot, you type box plot. And these are not things you need to remember, but I think knowing that they all start with geome really helps. So then in the future, if you want to plot something, you know to look for a geome and then something, and then it's easier to Google. Okay, so we had our plot, and then we had geome point, but then let's say we want to actually add in some color. So you can color it by any column. So again, this will be part of the aesthetic mapping because it's mapping a color to a column. And so you can then put in any column and it'll color the points based on what's in that column. And there's several aesthetic mappings. So I've talked about X and Y and color. You can also change the fill, which is a different way to color the points, the size of points, and the transparency. And again, I'll show you examples of all of these. And I keep saying that this is additive. So this is what we were just talking about. You can also add on different themes. So this theme will just change the background color. You can add on labels. So again, you always have to use a plus between each layer that you're adding on. You can label it with, you can type in whatever you want, type in good title for your title. You can facet it by, again, adding a facet layer. Yeah, and that's generally how you'll build your plot. So as I'm going through the example today, I'll kind of try to build it at this point where you look, like you plot the initial data and then you add on things. And this is how I do plotting myself. I'll always start with the basics and then see like, what do I want to color by? What do I want to like add as the label? And then finally, I want to just put this up in this plot so that in the slide deck, so you can click on these links if you want. Here's two just like good resources. And then there's two data frames I'm going to talk about today. One is Iris and one is RNA-seq. And a lot of examples I got came from these more comprehensive examples. So if you have questions and want to look at a data frame after this, I'd also highly recommend these tutorials. So again, after this, I'm going to go back to the notebook and show you some examples. And again, our big goal is to hopefully understand these building blocks in the grammar and some of these things that I've just introduced. Thank you. And then this is the link to the Colab tutorial. So if you'd like to follow along, you can do this. Otherwise, I'll be sharing my screen so you can watch. And I believe this is also posted on the website. So if for some reason this doesn't work, you can go to the website. With that, I will switch to sharing my notebook. That show up on the Zoom?
Yeah, it's
Zoomed in. Is that better? Is that still readable from in this room? Okay, perfect. Great, so this is the notebook. So the first thing we have to do is install tidyverse, which is this overall package that contains ggplot. So by installing tidyverse, you're also installing the ggplot package. And then we're also just going to load this datasets package that just has some of built-in datasets that are nice and clean and easy to play around with. And so, like I introduced in the PowerPoint, Iris is one of the datasets that we're going to look at. So it has 4 measurements from 50 flowers, and we can learn more if we do question mark, which is a nice R trick. And this will explain more about the dataset. Also, we can just look at it ourselves. So we have these 4 measurements, so petal length, petal width, sepal length, and sepal width, and then each species. So each row is 1 iris, and then you have multiple measurements. So this is the data set that we're going to start with, and this is the plot that I had introduced in the PowerPoint, where we have iris is the name of the data frame, and that's just built in that data sets. We set our 2 aesthetics as those 2 columns, and then we added geome points. Again, here, our x-axis, our y-axis, and the points. Like I mentioned, this is not a super informative plot because you can't see what the actual distribution is. So you can change the geome. So one example is to make this a box plot. And then we have a box plot. And maybe you want to have the box plot as well as points. Because it's additive, you can always add more onto it. So we can add on geome point as well. And then now we have the box plot with the point layer on top. And what is kind of fun, but can also be tricky, is that it plots things in the order that they come. So here we plotted the box plot first and then the point. If we were to switch the order of these, see what happens. And this is, I think, the best way to use ggplot too, is just playing around and seeing what happens. But if you plot them in this order, then it plots the points and then the box is on top. So then you don't see all the points. So that's always something to think about as you're plotting, is like, what do you want to see layered on top of what? Okay. So there we have our basic plot, but you might want to color this. So for example, you want to visually quickly see the difference. So that would go into our aesthetic mapping. And we can first try to do color, and then we can choose whatever column we want to specify this. So let's try species. And then when you do that, now it's colored by the species. So because we put this as the aesthetic mapping, it's basically using this label for every point as the color point. And so there's this color one. There's also fill, which is another way of coloring it. That's going to now fill in the box plot, whereas color was coloring. And then I mentioned labels. So it will label it by the columns that you give it. In this case, they're fairly intuitive, but if they're not, you can always add your own label. So that's another layer. Yeah. Yeah, so they're not colored because it's doing fill. So they're specified by color. So if you do color, then it will color the points.
One thing to note is that when you put your arguments in the ggplot, every layer inherits that same set of arguments. So if you, you have to make sure that everything there applies to your layers afterwards.
So if you wanted to like just color the points, you could do an aesthetic mapping within that like geom point. And then if you do equals, then there's no mapping overall, but like just that color mapping just matches it. You could always combine them. So if you wanted to do like an overall fill, as well as the point mapping, it works, but then because you kind of lose some of them. But this is like, you can start playing around with it. There's lots of fun things to do. My favorite thing to do with points as well is you can do position equals jitter. So now it keeps their y point is the same, but it'll like vary their x. So whereas we had overlapping points, now you can see that they're all spread out. And this is also a flexible function. So you can say width equals like 0.1. And then instead of spreading them across the entire width of that box plot, it's just slightly varied. So you can start seeing where the overlapping points are, which is data that you lose if they're all like layered on top of each other. And I think, oh yeah, so then labels. We want to add our own labels. That will be its own layer, it's called labs. And then you just type x equals and then whatever label you want. So here you just say like iris species. We'll say people. You can also add a title. We'll say what is this? And then now you've changed your titles. These titles look kind of small, so you can add in another layer for theme. And then this is where you can adjust your text. So here you can say text equals element text. Let's say 20. So you have to say the text is this element, and then within that element, it has this feature of size. And then now our text is bigger. So these are some of the examples. Again, a lot of these I always have to look up. So sometimes I'll forget like text equals element text or like where that goes. So I think it's kind of useful to understand this layering. And then again, I think it's kind of useless to try to remember everything. But once you know the structure, you can start thinking of like, what things do you want to add on and then add them as layers.
One of my favorite theme arguments is live in position.
Yeah. Yeah. So for example, if you want no position, you could say none, I think. Yeah, and then that should remove the legend. And then yeah, So then now move the legend to the bottom. I assume top would do the same. See? Yeah, so then it puts it at the top. So then once you have the features, you can just move them around, which actually can become really fun. It can also become, can drive you crazy.
Yeah, make your own favorite and just apply
Yeah.
it.
Yeah. Oh yeah, another fun thing I like adding is this theme BW. And I think that this makes plots look nice because then instead of the background being gray, then it makes it just black and white. So you can really focus on what your plots are. So that's something I almost always add onto plots just because I like it. A lot of these are kind of aesthetic choices you can make. It becomes fun and it makes you like, you can make things exactly the way you'd like them to look.
So can I ask about this
Yeah.
theme? You can add theme, black and white, and then additional theme?
Okay. Yeah, so this is just setting like the background theme, and then these are like customized changes on top. Yeah. And yes, please interrupt with questions at any point. And this isn't the only one. So I think there's also theme gray. And then that will make it more, I think that's the default. So there's lots of options, and then you can adjust it on top of that. And then skip this next cell because my goal was to build from this cell into this cell. We go to the next one. This is the exact same plot we were looking at, but now I added, so we were looking at geom box plot. I also wanted to show you what, so that's geom box plot. And then you can also do geom violin, which shows very similar data, but now the width of the violin is showing like the density of the plots. So that's another way to visualize the same type of data. And then, oh yeah, I showed this example of coloring by species instead of filling. And so those were kind of my box plot examples. Were there any questions on that before I move on to a different type of plot?
I have
So then with that, I also wanted to show some scatter plots. So this is more of your traditional scatter plot. Here I wanted to compare the length of the petals to the width of the petals. So really the only thing that I changed here was instead of X being species, X is now petal length, just the X axis, and then Y is the petal width. And then we add on geom points. So again, this is kind of the base of the plot. And then I'll show you how I get to this. So you have this base, and then we can add on the, whoa, what I did. Okay, you can add on theme we had before, just to make it look a little cleaner. And then here we can see that there's kind of this spread and maybe different clusters, but we don't know what these are. So one nice thing might be to color this by species. So you can see if these distributions are clustering by species. So we'll put that in under the static mapping and apply that to the entire plot. Color equals species. So then now we can see that, oh, this is one of the species, and then the two others are up here. And it's kind of fun is in addition to coloring, you can also map the shapes. You can do shape equals species. It's a little small, but now in addition to the red ones being the Setosa species, now those are also the circles, and then there's triangles, and there's also squares. So you can, in this case, there's only really one variable we'd want to color by, but in theory, you could color by one variable, add, like change the shapes based on another variable to show multiple types of information in one plot. That should, oh yeah, and then something else. So you can just change overall the size of the points. So because this is not within an aesthetic mapping, this will apply to all of the points. So I want size equals like 3. So that will then just change the size of all of the points. If you put this inside of an aesthetic mapping, you could then make this be species. And then their size would vary based on the species. So that's kind of the difference of using a mapping versus using just one assignment for all of the points. So I like using one size for all of them, and then you can just make it the way you'd like it to. Yep. So that's the plot I was hoping to get to. And so that's all kind of looking at plots, but sometimes you also want to ask questions about this. So for example, these look like they're correlated, You could run statistics and get your own like regression lines. But you can also just plot that. So if we add this geom smooth and then method LM, which is like for linear method, I think, or linear regression, this will then add on this linear line. So it's just fitting a y equals x line, which I guess the warning of this is that it doesn't automatically give you the slope and intercept. So you'd want to run your own regression to get those values. But it can be a nice way just to see if you have a lot of points to see if there actually is a pattern, and then you can explore that more with your own regression lines. I don't want to go too much into the statistics of it here. But you can use that to just plot a point or plot a line. But let's say you have your own idea. So you think maybe these are like directly correlated, or that one of them is half the value of the other. You can add on your own lines. Let this out for now. You can add on, so a line is going to be an AB line. So geom AB line where you specify intercept and then the slope. So for example, so we have this idea that maybe there's an intercept at 0 and a slope of 1. You can plot that. And then that will now just show up as a line with the parameters that you gave it. It's clearly not 1-to-1, but sometimes this can be nice if you are expecting it to be 1-to-1. So you can show your expectation and then show the data, and it's very clear that they're not the same. So for example, maybe the slope is 0.5. You can see that then it gets slightly closer. And of course, you can always like fit a fitted line and then see how the fitted line varies from your like expectation. Yeah.
Is it possible to show the equation of the line on the line, or do we have the?
Yeah, it's a good question. I was actually, yeah, I was looking it up because I thought it would be really nice if it printed. It doesn't look like you can. It looks like you have to fit the regression separately to get it. I was really hoping there would be, because I was hoping to print that. But yeah, from what I could tell, you can't, but it might be possible. And that. So that's fitting an AB line. What can also be nice is to fit, or not to fit, but to just plot like a horizontal line or a vertical line. So let's say you're interested in everything that's above 1.5. That would then be this layer called geom H line. So there's H line and V line. So it's a horizontal line or a vertical line. And then the AB line I think of as like you have your A and your B coordinates. If you do your H line, you then want to specify your Y intercept. Yeah.
Sarah, there was a question that said that
Yeah.
you mentioned earlier, but somebody wants to know if there's a difference between using the title or the title less.
Yeah. That's a great question. I'll show the lines and then show that. So here, the H line set our Y intercept, and then it creates horizontal line at the intercept. And you can change the line. So for example, if you want this to be red, do color equals red. That will change the color of that line. You can also change line type. Yeah, dashed. And then now you have a dashed red line. So this can be nice for publications if you're interested in like it's above this. So you can like show it so then someone can visually look at your graph and know what's above, below. And then if you want to do a V line, it's the exact same thing, except then you give it an X intercept. And here we could say like 2, and then it will create a vertical line at that intercept. And then for the Zoom question about the difference in titles, so I think it's the exact same. So if you do labs and then title equals title, get rid of this label. Maybe the title, and then you could also do GG title, and I think should also work. So I think it's the exact same mechanism. There might be slightly different defaults, I'm not sure, but either one works. I don't think you can do that here. So if you do like X equals, yeah, that doesn't work. So I think the GG title only applies to the top, and then within labs you can like specify all of them.
So then
I think. Yeah, so within labs, this one, and you could do X equals X. And then that should work. Yeah, and change the x-axis. Yeah. Mhm. Yes. So I think that's with geom text, I believe. So you could say like y equals 1.5, and I believe you give it points, like x equals 2, y equals. See if this works. It didn't work. Maybe
Well, maybe position.
position.
Yes.
Oh, yeah, I think you're right. Okay, so then we could do 2. There is a way. I know it's with geom text. Yeah, you have to. Is a way. I'm not exactly sure how it is. I'd have to look that up for you. But I know within geom, maybe it's geom. That's something I would Google. So I know there is a way to do it where you can like give it the title you want and then like specify the coordinates. Just don't remember exactly. Great. So that was plotting, and so we all remember this is kind of where we left it, where we had a scatter plot of the petal length and the petal width. And now all of the species are on one plot, but they're colored by their species. But you also might want to see each of these as separate plots. So maybe you think there's something different going on in this species versus this species. And so a great way to do that is by faceting. Now, so within the plot that we have, you can then add this facet wrap, and then you do the tilde, and then the column that you want to separate it by. So in this case, if you separate it by species, then based on the species that you give it, it'll create like a separate plot for each of them, and then the top labels the species. So that's how you can create this like multi-paneled one. Within this plot, this is basically the same plot that we were just looking at, but what is cool is you can change the scale. So if you do scales equals free, then that will adjust the scale for each plot. So then that could allow you to see what's going on here, what's going on here, which can be nice if you think there might be slightly different patterns within each data set. That's a nice flexible option. And then I did want to show how you can manipulate data frames to then plot it. This starts getting a bit more into the tidyverse stuff that Monica had talked about before. But if iris is your data frame, and then let's say you want to create this plot, so you have a separate plot for petal width and sepal length based on the species. Because everything is given in columns, you have to adjust the data frame a bit so that these variables are in the columns that you want. So then you'd have to have a column that will either say petal width or sepal length, and then another one that says name. So I'll kind of explain how this works. Again, if you have questions on how this works, I would suggest looking at Monica's tutorial, or just more of like our data frame manipulation, which is a bit more of what's happening here than the plotting. But basically, we take the data frame, we combine these two columns, so petal width and sepal length, so that then they each have a value, and then there's a name. I can like draw what this looks like. Now we have like, maybe this will catch up, but you have like the same data frame that you had. Now you have name, and we'll say like sepal length, width, and then you have your value, like 2, 3, 4. And then each of these will additionally have like what's their species, and all the other information. So now this is the column that we can facet on. So that then you are plotting X as species, Y is now this value, and then we're faceting it on this name. So each of these names now becomes one of the subplot titles. And so then you can see like the ratio or the relationship between species and value within each of those categories. So this is kind of the same faceting, but with some data frame manipulations.
Professor,
Yeah.
I looked it up
Mhm.
because view text will work for like points, but if you just want to slap like a label on the plot, it's annotate.
Annotate. Okay, let's try that. So, okay.
And then if you can use the X and Y.
Okay. It's like,
When you use something, view text, it's
Uh-huh.
expecting like an aesthetic mapping for your.
ah, is it X and Y?
View.
View annotate?
Oh, and what text are you?
This label?
That's the, yes.
Geom annotate? No. Okay, no, thanks for looking at something like that. That makes sense. Thanks. Okay. So we're here. And then there's just a few more examples of iris. And then if there's questions, we can look at them. Otherwise, we can go into like an RNA-seq expression data set. One of the final things I wanted to show is that sometimes you'd want to plot a value that isn't in your data frame. So far, our data frame has these lengths, these widths, but what if you want to know the ratio of the petal to the width? So one way you can do this is you can just input equations within the ggplot framework. So for example, ratio of length to width, you can just type y is now petal length divided by petal width. So it will divide those columns by each other to now give this new value that you're mapping onto each x point. You do that, now you get this nice little ratio. But I personally think it's a fast, easy way to look at it, but I think a more reproducible way is to add this as a column into your data frame, which you could do. I had to create a new data frame. So now my iris equals that same iris data frame, but I'm adding on this new column. So length width ratio equals my iris petal length divided by my by the petal length, yeah, divided by petal width. And then you can now plot that ratio, and now you have the exact same. So just add things, and as long as it's a column, you can input it into this framework. And then, okay, this is like my favorite part, is that you can change all the colors and it's very flexible. So I looked up, and there's this website that has like the names of all the colors, which I can show you. Very fun. Basically, this website gives you all the possible colors and then what their name is. So I just found a few that I liked. So I wanted to try now coloring it with deep pink, chartreuse, and cyan instead of their default ones. You can create a color palette. And then we're now doing the exact same plot where we're plotting that ratio. And now you just add in this scale fill manual. So basically you're manually applying values to that fill mapping. And you can input the values that you want. So in this case, my palette. And then you plot it. I didn't run this plot. You run this. And then run this. And now it has the colors that you want. That's fun. So then you're not just limited. And also this can be nice. Everybody always uses the default, so it can make your plot stand out a bit more. Yeah.
I figured out what you're missing when you're doing.
Yeah.
What you're labeling. So in the annotate.
Oh, okay. Okay. So annotate.
So if the first argument is text,
Yay. Okay. Thank you so much. Okay. There's your answer. So you can do annotate, text, I guess.
yeah.
Are there other things you can? Okay. That's good. You can give it X coordinate, Y coordinate. And if you wanted this to be like slightly above the line, you could do like 1.7 and make this like 0.2. And then, oh, you could adjust it and then try to get it to like right there as some visual place. So that would be the answer. Label. And then that would apply to like every point. Oh, so like, for example, if we did geom text and then do the aesthetic mapping to species. Wow. Okay. That's something to play around with later. But thank you, Monica. That's
Yeah.
a good solution. Yeah. Okay, so here we have our colored plots. So this is all great. So far, I've been looking at plots within this notebook, but if you're trying to like share this with somebody or actually write a publication, you want to get these plots like saved onto your computer as a PDF that then you can like upload. And so there's a few ways to do it. The way that I like doing it is just with PDF. So you do the PDF function, you specify the name and the path. So I'm just going to call this myplot.pdf. You can give it height and width, which can be nice to adjust. And then you just plot it, and then you do dev.off. So basically it's opening up this PDF, and then as you plot this, it's putting, instead of like showing you the PDF or showing you the plot, it plots it to the PDF, and then it just closes the PDF. So if we run this, then that should, yeah. So now we have myplot.pdf. You could then download, then show up as a nice PDF that you then have on your computer, and then you could drag and drop that into a PowerPoint or a presentation or a publication or whatever. So that's kind of how you get from plotting to then something that you can use moving forward. And I think there's also a GG save. There's a few options to do this. This is the way I work that works for me. I think is a pretty flexible option. So then, for example, if you wanted to change these, you can make width like 8, height 3, and then download this. It should be different dimensions. Yeah, so now it's changed the dimensions of our PDF. So you can kind of change the way that it looks based on the space you're trying to fit it into or what you want it to look like. Great, so those were all the examples I had with this iris data set, which is probably the most clear. I also have a larger RNA-seq data. Does anybody have questions on what I just talked about before I move on? Okay, nothing on Zoom. Okay, great. Yeah, then I'll show you a larger data set. And this also like reads in data, so you can see how that would work if you had your own data set that you're wanting to load in a plot. So here, this first one step is just downloading the file. This is an online, this is data from an online publication. And so basically this is just downloading that file. And then here I am reading in the file. So this command just saved it as a CSV. If you were doing this with your own data, you'd likely have your own file that you're starting with, and then you can just read it in as a data frame like this. And this data frame that we're going to be looking at is, I mentioned RNA-seq, and they're looking at transcriptomic changes within the central nervous system after influenza infection. So the data that we have, they have mice that were infected with influenza, and then they have time points of 0, 4, and 8. So different mice at different times, because if you take out their cells, you can't really let them keep living. But basically you have different mice, different times, and then they're infected at time 0, and then you can see it later. So I want to see what this data frame looks like. You have each gene, and then the sample, so that would be the mouse that it's from, if it was infected or not, the mouse ID. Oh yeah, the time. So this is at time 8, so it's infected, and then I think it's 8 weeks. I have to read the paper. But then this is the product of the gene, and then there's also some different gene IDs, and then the type of gene it is. So this is a protein coding gene that's involved with this amino acid level. So basically, you have an expression level for each gene, and then for each mouse, and then each mouse has its own characteristics. And so I think a first question one might have, if looking at this, is just what is the overall expression level? So here I am plotting a histogram, and this is one of the only genomes where you don't have to specify a Y, because it's counting the number of occurrences of each expression level. So basically, every expression is binned. Yeah, so you have all of your binned expression levels, and then count. So that's like the number of mice that it occurred which is nice. And you can also, oh yeah, so you can change this to density, which is another one where you don't have to specify the y-axis. And it does a very similar plot. We can see like, it's basically the same plot, but now it's like in a smooth line. If we make it to histogram, one thing that stands out to me is that this is largely skewed to the left. So something I was curious with is what if you plot the x-axis on a log scale, so that you can actually, it'll kind of be 10 and then 100, so you can see what's actually happening at the lower level. So you can change the scale of the x-axis. So here, if we put it on a log 10 axis, I don't know why that didn't work. This morning. There you go. Yeah, so there we're plotting the same expression column, but now it's on a log scale. So you have 10, 100, 1,000. So you can kind of see more of like what's happening. So that's kind of a fun trick to have you're visualizing, so that you can like change the axis scales. Yeah, so I added in this. So if you don't add that, it does the plot we were looking at. And then if you add that plus scale x log 10, so it's changing the scale of the axis, x-axis, to a log 10 scale. Yeah, so then, and you can do the same for y. I don't think would make any sense in this data set, but you could, and then the y-axis is now clearly on this log scale to kind of visualize it in different ways. Which is great, but this is still like at every time point within all the mice. So this is probably, did you have a question?
No.
Oh, okay, sorry. So probably the last thing that we'll look at is what if you actually want to look at expression for each gene. So here we'll look, X will be the gene, Y will be the expression level. As a starting point, this is not a very informative plot yet because genes, there's a ton of genes, you can see their expression. So you can see there's variation, but we still don't know what time it is, whether they're infected or not. So a nice thing to do could be to just color this, actually. So we'll do an aesthetic mapping and we'll color it by time. So now, yeah. So now it's colored by time and it's doing a heat map. I want these to be discrete time points because we know that there's 0, 4, or 8. So we do character. Now it's changing these from numbers to characters. So it should just plot them as 3 different. So now it's just plotting, or it's coloring by 0 or 4, 8. Yeah.
So we have just a couple minutes
Yeah.
left. I want to make sure that you're okay with staying with lots of questions.
Yeah.
Should we,
Yeah, I'm happy to stay after.
yeah, so we just have it for 1:30, but
Okay, yeah, I'll show maybe like one final thing and then, yeah, I'm happy to stay after and, yeah, ask questions. And I think, I don't know if any of my contact information's on the
maybe
website.
share
Yeah.
that. I think share it on Zoom. Here we go.
Yeah. That's fine. Thanks. I guess the final thing I might show for this is if we instead want to facet, and we'll facet it by the time. So then, yeah. So now it's colored by the time, then also you have a facet for each time point. And you can instead color it by, it's like a gene. I don't remember what it was called. There's some column that said the type of gene. Gene biotype. So we color it by gene biotype. Then you can see almost all of these are encoding, which makes sense, but you can still see at each time point, then you have a visual of the expression. You can start seeing the gene biotypes, and then this is starting to give you a bit more information. And if this is your own data, you can start filtering it more, looking at the top expression, looking at fold change differences. But this is kind of an example of if you have a larger data set, how to start breaking it into pieces and get to something that's like a little bit more clear. Yeah, and I think I'll end with that. And again, happy to stay later and answer any questions. And yeah, feel free to email me and get any materials. And thanks for coming. Yeah.